I'm going to discuss about another immunodiffusion technique, which is also similar to Octoloni immunodiffusion technique. There also the diffusion of antigen and the antibody took place radially, but you demonstrated a simple precipitin band. Here, you are going to demonstrate the formation of precipitin band in the form of circles around the well. So, the radial immunodiffusion technique is also named after a scientist, and it is called as Mancini's method. Mancini's method. For this, you would require a large glass plate as shown here, a large glass plate. Once again, clean the surface of this glass plate with a tissue paper moistened with alcohol. After you have cleaned the surface, you would require now the same materials that you had used for the preparation of slides. That is, you will require agarose of very good quality and the suitable buffer. And for the demonstration, you would require soluble antibody as well as antigen to demonstrate the radial immunodiffusion. Rest of the other materials that are shown here are same, like pipette to pour the agarose onto the slide, the rubber bulb that is for punching, which is having the improvised micropipette tip whose tip is cut, and you need the pasture pipette. In case if you want to use the pasture pipette, you can use the pasture pipette for pouring the agarose on the slide. And the needle you would require to remove the agarose from the wells in case if it has not come out from the wells. Now let us see how the slide is prepared. In the same manner, you take the required quantity of the agarose. Each glass slide of this size, would require about 15 ml of the molten agarose. The glass slide of this size would require 15 ml of molten agarose. You can measure that much quantity of the buffer. If you are preparing more than one slide, measure that much quantity of the buffer, appropriate buffer that is given with the kit, and take the measured quantity of agarose. You would require about 1.5 to 2% agarose and gently melt it in the water bath by keeping it in a low heat. And once the agarose melts, it becomes totally transparent without any particles. So take this molten agarose, the required quantity, into a beaker. Okay, take the required quantity of that agarose into a beaker. Then allow it to cool down to at least about 40 to 45 degrees so that you will be able to hold it in your hand. It should not be hot enough, because if it is too hot, when you add the antibody into this agarose, the antibody may get denatured. So to avoid denaturation of the antibody, see that the agarose is cooled sufficiently but not solidified. About approximately 40 to 45 degrees, you should be able to hold it between your fingers. The beaker should be so hot enough that you should be able to hold the beaker. Now, take the antibody that is to be used. Required quantity of the antibody is pipetted out using the micropipette, which is provided with the kit. And put this required quantity of antibody in the agarose, which is cool enough, that is about 40 to 45 degrees. And after adding that antibody into the molten agarose, gently rotate the beaker on the horizontal surface. But do not be very harsh. The air bubbles should not get trapped in the agarose. Gently rotate it. After gently rotating, take now required quantity of the molten agarose which is already mixed with the antibody into the pipette. As I told you, the glass plates of this size would require 15 ml of the molten agarose. And once you pipette the required quantity of the agarose into the take the agarose in the pipette. Now pour that agarose onto the slide which is kept on the horizontal surface. This will also form a 3 millimeter thick agarose gel on the glass plate. Second slide is being prepared. Okay, sometimes a air bubble may come. Now use a aperture pipette, burst the air bubble or remove the air bubble by sucking. The slides with agarose which is already mixed with the antibody is ready. Allow the agarose to solidify. Once the agarose solidifies, punch the wells. As I had mentioned earlier, to enhance the solidification of agarose, once the agarose is solidified, you can keep the slide in the moist chamber as I had shown you earlier. By keeping the slide in the moist chamber and then covering it with a lid and keeping it in the fridge, the agarose hardens faster. But do not handle the slide when the agarose is molten. Once the agarose is hardened, by keeping it in the moist chamber, you can punch the slides as shown earlier. Then to the punched wells, you have to add now standard antigens which are labeled as S1, S2, S3, S4, S5 like that. The standard antigen is nothing but known concentration of antigen corresponding to the antibody that you have already added. You have already added the antibody into the agarose and you have allowed the agarose to solidify. Now into these wells, you will be adding the known concentration of antigen in an increasing order. Then into the U1, U2, U3 unknown concentration of antigen that is to be tested, and to find the concentration of antigen in that unknown sample, you will be adding into these wells. Unlike in the Octoloni technique, where you will not be able to determine the concentration of the antigen, here in this technique, radial immunodiffusion, you can find out the concentration of the antigen from the known concentration of antigen. So, known concentration of antigens are loaded into the wells which are labeled as S1, S2, S3. Same amount, like each well would take about 10 microliter of the sample because the depth of the well is approximately 3 to 3.5 millimeters. See to it that when you load the sample, it does not overflow over the well. Overflowing should be avoided. After adding all the standard or known concentration of antigens in the respective wells, you may load the unknown samples into the wells that are labeled as U1, U2, U3, like that. Each time, do not forget to change the tip. Why? Because the concentration of the standard antigens was different. Now you will load the unknown sample whose concentration has to be found out. The antigen concentration in the unknown sample has to be found out. It could be even the serum from the suspected patient. Load it in the respective well which has been labeled as unknown. If you have more than one unknown sample, you may load in each respective wells the unknown samples. At the same time, see that you have changed the tips each time when you are loading the new sample. Please take care that the tip of the micropipette doesn't touch the surface of the agarose and tear it. You have to be very careful while loading. Support the tip with your left hand so that you don't tear the agarose gel. Once the samples are loaded, once again prepare the moist chamber. The moist chamber may be there readily, and see that the moist chamber has sufficient amount of moisture to hold the slide moist. Here I am showing you once again the preparation of the moist chamber by making the bedding of the filter paper, which is moistened with distilled water, and the surface is pressed uniformly onto the bottom of the box. Do not flood it. To the moist chamber, now the slide which is loaded with samples is transferred. You transfer into the moist chamber, cover it, cover the lid, and then put it in the fridge. Keep it in the fridge and the lower chamber overnight. Once again, this reaction takes 24 hours for the diffusion of the antigen and the antibody. So after 24 hours, take out the slide and observe. You will see the precipitin bands like rings around the well. And in this slide, you can already see there is difference in the size of the pristine bands. One can make out from the size of this pristine band on the extreme left-hand side on top that the concentration of the antigen is very, very low. As the concentration of the antigen increases, that is the standard antigen we have loaded in increasing in concentration, the size of the ring will also increase. The size of the ring will also increase. So, larger the ring size is equal to more the concentration of antigen corresponding to the antibody. Now, measure the distance. People say sometimes diameter, but it is actually not diameter. Measure the distance of the ring, the peripheral part of the ring, to the periphery of the well. When you measure the distance of the ring from the periphery of the ring to the periphery of the well, it is the distance, because you are not taking from the center of the well to the periphery, it is not diameter. So once you measure the distance either in millimeters or centimeters, note down the readings. For the increasing concentration of the antigen, you can note down the readings, and then for the unknown samples, you can note down the readings. Now here in this slide, you can see the concentration of the samples. Like sample number 1 is supposed to be standard, which has a large ring. From the periphery of the well to the periphery of the ring, the pristine line, that is the periphery, you can call that distance. You measure that in millimeters. One can keep this transparent slide on a graph paper and measure each division of graph paper as 1 millimeter and find out the distance in millimeter. Or one can keep this on a steel scale which is graduated and find out the distance. Here in this diagram, you can see unknowns are labeled as U1, U3, U2 here. The distances or the ring size is different for different unknown samples. In this petri dish also, they have demonstrated radial immunodiffusion. Because this technique is called as radial immunodiffusion, because it radially diffuses the antibody that is there in the agarose, and antigen that is added in the well diffuses in all directions radially and comes in contact with the antibody by developing a pristine ring around the well. So therefore it is called as radial immunodiffusion. So once you find the distances, you can plot a graph. Okay, the concentration of antigen, you take it on the x-axis, because you have been given the known concentration of antigen. You can take it in the increasing order in the x-axis, and then it is mentioned here as diameter, but actually it is not diameter, it is distance. So the distance in millimeter can be measured and plotted. The distance in millimeter is plotted against the concentration of antigen, and you will get a linear curve like this. And once you have prepared a standard graph, the unknown sample whose distance, the pristine line distance was measured, can be found out from the standard graph, and the concentration of the unknown antigen can be found out in this manner. Unknown concentration, the distance is matched with the standard graph, and unknown concentration can be found out. Say, as in the previous example, you had 3 unknown samples of 3 different sizes of the rings around the well. One can find out the concentration of each of these by comparing with the standards that were already loaded, and thus the ring size of the standards is dependent on the concentration of the known antigen, the standard antigen, and that can be matched with the unknown, and the concentration of unknown can be found out. Therefore, radial immunodiffusion which is called as Mancini's method, can be also used for finding out the concentration of antigen. Suppose if you had used the antigen in the agarose instead of antibody, you can measure the concentration of the antibody. The technique can be reversed. In this technique, I had shown you that we had added antibody into the molten agarose and known concentration of antigens were taken as standard and unknown samples were loaded and unknown samples concentration can be found out. Similarly, if you use the antigen in the molten agarose, you can take the known concentration of the antibodies as standard and then unknown concentration of the antibody also can be found out from this technique. So, the technique can be used for finding the concentration of antigen or the antibody by using radial immunodiffusion method.